Happy post-Valentine's Day. Thank you for joining us today as we present, along with our friends from the Council for Federal Cannabis Regulation and the FDA, about the endocannabinoid system and current implications for both research and regulatory science. Before we begin, we wanted to cover a few housekeeping items. If you have any questions during the webcast, you can submit them through the Q&A widget at the bottom of your screen. Your questions will only be seen by the presenters. And you can find answers to some common technical issues located in the help widget at the bottom of your screen. And with that, I'm Jack Jacobson. I'm an advisor at Thompson Coburn, and I'm one of the Council for Federal Cannabis Regulations advisors. I'm incredibly excited about today's webinar, focusing on the endocannabinoid system and how federal regulators are digging into the related science and research and how it informs the regulatory process. With that, I'd like to introduce my co-host for the series, Sarah Chase. Sarah?
Well, hi, everyone. Good afternoon. I'm Sarah Chase, the Executive Director for the Council for Federal Cannabis Regulation, and it's my pleasure to welcome you to the latest installment of CFCR's webinar series. So today I'm thrilled to introduce you to our four brilliant guest speakers, each a subject matter expert with extensive experience and knowledge. And I encourage all of you here today to ask a lot of questions. Jack and I will try to get to as many as we can and to work them through and to make sure that this is a nice, good, informative afternoon for you. So without further ado, today we're joined by Dr. Cassandra Taylor, a chemist at the U.S. Food and Drug Administration, Dr. Dan Mellon, and Dan, I hope I get this right. You are the Deputy Director, Division of Pharmacology and Toxicology for Neuroscience, Office of Neuroscience, Office of New Drug Center for Drug Evaluation and Research at FDA. I did not memorize that, but hopefully I wrote it down. And we also are joined today by Mara Gordon, co-founder of Aunt Zelda's and a member of our medical subcommittee. And from CFCR's Science and Regulatory Affairs Committee, please join me in welcoming Dr. Vicki Seyfert-Margolis, who's chairwoman of that committee and the CEO of her own company called My Own Med, and Dr. Reggie Gaudino, co-chair of our Science and Regulatory Affairs Committee, and also chief science officer at Front Range Biosciences. So welcome, everyone. Dr. Taylor, perhaps you could start us off by introducing yourself.
Sure. Thanks so much, Sarah. Hi, everyone. My name is Cassie Taylor. I'm a chemist here in the Center for Drug Evaluation and Research at the FDA, and I work on the Botanical Review team. We specialize in the review of all submissions for botanical drug products. We are part of the integrated review process here in Cedar that includes folks like my colleague, Dr. Mellon, as well as our medical and our chemistry, manufacturing, and control colleagues. And I serve as a subject matter cannabis expert here within Cedar. Thanks, Sarah.
Thank you, Cassie. Dr. Mellon, welcome, and maybe you could tell us a bit about your role within FDA.
Sure. Thank you very much. As Cassie mentioned, I also work in the Center for Drug Evaluation and Research. I'm a pharmacologist. I work with a team of pharmacologists and toxicologists who evaluate the non-clinical safety information that is submitted for various investigational new drug applications, and ultimately, hopefully, at some point in time, NDAs as well, to try to help understand the safety profile of various compounds that people like to investigate or wish to investigate for a variety of applications. And ultimately help guide the medical staff in terms of what types of things perhaps we may need to monitor for, and guide companies on development programs as if they wish to try to pursue a marketing application. So we have a large group of people who've been working in a whole host of areas in the world of neuroscience, including some applications that, of course, with the interest in cannabis and cannabinoids.
Excellent. Well, thanks, Dr. Mellon. Mara, welcome. Maybe you could tell us a bit about your background.
Hi, thank you. I appreciate being here and being included in this very interesting webinar. My background is process engineer. I started collecting data around patient usage of cannabinoid-based medicine integrated in with their regular care well over a decade ago, and have created many formulations, have done clinical trials and preclinical research on some of the findings that we've had, and released a software platform that is now being utilized by physicians to better guide their patients with cannabinoid-based medicines.
Well, thanks, Mara. Vicki, also, thank you for joining us again. You were on our first webinar when we debuted long ago, and thank you again for being here. Maybe you could tell us a bit more about yourself. Hi, sure. I'm Vicki Seyfert-Margolis. I'm the founder and CEO of my company, MyOwnMed, which is a digital technology for health workflow optimization and data integration to support clinical research studies that are decentralized, that is combining direct from patient data capture and assessments that patients are doing in a decentralized fashion with in-clinic encounters to support hybrid clinical trials. We also are working on optimization of workflows in outpatient care settings. Prior to that, I was the senior advisor for regulatory science and policy to the FDA commissioner during the Obama administration from 2008 to 2012. I worked on a number of issues, including digital health, complex products, reinvigorating regulatory science, and several major policy initiatives as well. I'm an immunologist by training and have many, many years of clinical research experience in novel clinical trials with novel products.
Thanks, Vicki. And Reggie, great to see you again. Maybe you could tell us about yourself and your work as a molecular geneticist. I think you're on mute, Reggie.
Hi, I'm Reggie Gaudino. I'm Chief Science Officer at Front Range Biosciences. I lead a team of scientists understanding the role of the environment and gene expression in general on the outcome of products in cannabis. Do a lot of molecular genetic work. What that means is studying the DNA, looking at the expression of RNA and ultimately the phenotypes that we're looking at. That all goes towards breeding better cannabis cultivars that have a specific activity. You're looking for a specific compound or purpose bred for extraction or whatever like that. So just the ins and outs of cannabis, basically.
Fantastic. Well, thanks, Reggie. And just a reminder to the audience, please submit as many questions as you like. We will process through them as quickly as we can. So without further ado, let's jump right into the conversation. Dr. Gaudino, Reggie, perhaps you could take a moment to explain what we currently know about the endocannabinoid system. And I know this is a complex topic, but what do we know? Why is more research needed? Give us a benchmark.
So the endocannabinoid system generally, right, is a system that we've discovered because of cannabis and research with THC and CBD. But what it is it's a receptor-based system that acts to control homeostasis throughout many of the, if not all of the, biological systems in humans and other mammals. In fact, every organism that since we came out of the ocean has an endocannabinoid system. So, so it's really central to how our biological systems integrate together, right? And ironically, plants have evolved these chemicals as well. Endo means endogenous, cannabinoid, obviously, right? So but phytocannabinoids are very similar and play in the endocannabinoid receptor system. And so, what we now understand is that we know very little. The endocannabinoid system does its work through a series of second messenger systems. And that's where we really have to dig in deep to understand better, because second messenger systems affect a compound that triggers a second messenger isn't just triggering that one thing. Second messenger systems are in fact messenger systems. And so they then trigger a network of response. And without knowledge of what's going on there, we can't really call it a medicine. So.
Reggie, could you maybe explain a little bit too about what we know as the entourage effect? And
the entourage effect. So the entourage effect is generally how the phytochemical compounds that come out of cannabis, or any plant for that matter, right? So entourage is just basically referring to the chemical combination that comes out of when you do an extract or when you examine any plant-based or botanical, right? So the entourage effect in cannabis includes terpenes and cannabinoids, and why that is important is both terpenes and cannabinoids both interplay in the endocannabinoid system, right? And in fact, we do know now that it's not just cannabinoids that bind in the known CB1 and CB2 receptors. We actually now know that terpenes and cannabinoids will bind together in the receptor, and they interact in a way that changes how that receptor responds, right? So the entourage effect can mean a number of things. Generally, it's referred to in cannabis more so with terpenes, but and terpenes provide a lot of the setup, where then THC and CBD come in and are the finisher, if you want to look at it that way. So,
excellent. Well, Dr. Mellon, I've got a question for you. Now that we know sort of a little bit more about this, from the perspective of somebody who works in toxicology, and also you, Cassie, from chemistry too, what are some of the questions that we need to start asking about sort of these effects, the plants, what's happening within the human body? Sort of what's sort of top of mind when you start to look at the science behind this?
Certainly, I can go ahead and get started. I think this is a fascinating and a very challenging issue to really try to work through. And and as Reggie noted, the belief, presumably with any botanical, whether it be cannabis or anything else, is that the mixture itself of the various compounds that are present in the material is a little bit different than the isolated compounds. That certainly, very well may be true. And I think that's one of the reasons why the agency has established a very nice guidance that describes botanical drug development, because, if that is indeed the case, then that mixture may also have very different safety profiles. It may have different pharmacology. It may have different toxicological profiles. And as a result, one of the biggest challenges is trying to really bridge the knowledge that we have on those particular compounds and that particular mixture from one product to another, or trying to leverage data that perhaps may not have been generated with that particular compound. And I think that is one of the biggest challenges. You really can't have a situation where the belief is that the mixture is pharmacologically different and yet toxicologically the same. Pharmacology and toxicology are natural extensions. It's all about the dose, right? So I think it's very important to think about those things as anybody starts trying to think about how they might try to develop products of this nature.
And Cassie, feel free to weigh in there a little bit with your thoughts too as a chemist.
Sure, Sarah. Yeah, I can add on. So in addition to all the things that Dan was just talking about, what my team does is we actually look at it from the pharmacognosy perspective. So we look at the raw materials themselves, because the way that the raw materials are grown will change the chemistry of the plant. So if Sarah and I both buy a basil plant at the Home Depot, even though we both live in the DC, Maryland area, there's different growing conditions, right? So there will be slightly different chemistries. So that's actually very important for any botanical drug that you're trying to develop. It doesn't have to be cannabis. It could be turmeric. It could be green tea. And so my team really focuses on the control of those botanical raw materials. And Dan had mentioned that we have a botanical drug development guidance, but we also have a draft cannabis guidance that has quality considerations for anyone who is wishing to pursue drug development for cannabis or cannabis-derived products. And there's lots of resources there, because Dan and I can't cover all of them today, but I think the chemistry and the raw material controls are very important. And also the different routes of administration you have to take into consideration, how you're formulating things. There's a lot of processes and there's a lot of steps that you have to take into consideration if you're planning to conduct a clinical trial in humans. And so we have a lot of resources that are already available to folks. And botanical drugs, there are 2 already that are approved. And so one of them is Verogen, which is the green tea extract. And then we also have crofelemer, which is dragon's blood, but I think it's called Mytesi now is the new name. But there's already 2 approved botanical drug products, and we have lots of botanical drug applications that are here at FDA. So if anyone is interested, we have those resources already available to you. But mainly we're covering the raw material controls, the CMC, which is a big part of CMC starts with the manufacturing. So when you're taking your raw material and you're manipulating it in some way, let's say you're adding chemicals, you're adding some sort of heat, there's an exchange of chemical composition. That's really when the manufacturing starts to occur. And then you move beyond that with the farm tox and the clinical aspects. And there are other disciplines that can get involved depending on the complexity of your product. So it's important for folks to make sure that they look at those resources if that's what they want to consider. And we can talk more in depth, but I want to make sure we give the other panelists some time as well, Sarah.
Well, Cassie, I was hoping you might be able to dive in a little bit more into the process for assessing safety and efficacy of botanicals. And maybe that's included in those guidance documents that you've been discussing, but would love just an overview there.
So generally, the general process. So yeah, good question, Jack. So the general process is here in CDER, any drug submission that comes into CDER, there's a team-based review process. So if it's a botanical drug, my team would be involved. If it was an indication that went into Dan's division, he's in the Office of New Drugs, all submissions are based on the indication. So the clinical indication that, let's pretend Jack is the sponsor and he wants to study, some sort of a pain indication, he would have to be specific. It would go into the pain division. They would make a determination, and then the team would be created. If it's a botanical, let's say it's a cannabis drug. So my team would be involved, chemistry, manufacturing, controls, farm tox team, and then the medical team. Now, it also will depend on the phase at which Jack's proposing his study to be. Let's say it's a phase 1 study. So it's an early phase study, but then there are all these different factors that you have to take into consideration. This is all extensively outlined, not only in the botanical guidance, but a plethora of other FDA guidances for each discipline. But very high level, you need to take into consideration who is the population. Are you going to propose treating healthy patients, Jack, or are you proposing to treat patients that have a specific ailment? We will give different advice based off of that. Are you proposing to use something that is already an approved drug? Are you proposing to use something that has extensive previous human use and experience? Now, each of these things is taken on a case-by-case basis. So myself and Dan cannot give any specifics to any one person's drug development program because each one of those drug development programs is unique. However, you can take these themes, and as you move through your drug development, so from phase 1 to 2 to 3, and then eventually to your new drug application phase, the NDA phase, the requirements for FDA for safety and efficacy will increase. And the botanical guidance, I think, is a really good place for those who are just first interacting with FDA, especially on the drug side. Our regulations are pretty clear about how we look at these things. So just for everyone's knowledge, when you come to CDER and you have an investigational new drug or a pre-investigational new drug application, if you're suggesting to diagnose, treat, cure, mitigate some sort of indication, you are a drug and you're going to use it in a human. So you have to meet 21 CFR regulations for drugs once you come to FDA. So I just want to be really clear about that. There are lots of different regulations across FDA depending on your intended use. But if you intend to use something as a drug, you have to meet the drug regulations. So that's super high level. Dan, I don't know if there's anything else you wanted to add to that.
No, sure. Thank you, Cassie. I think you explained it perfectly. The challenge that we have, it's a great guidance document. If you're not familiar with it, you should take a look at the botanical guidance document. You can find it in any of your search engines. If you basically look for FDA guidance botanical, it should pop up pretty quickly. It really does describe the process. And I think the important thing that many people may not realize is that guidance recognizes the fact that there may be situations where there's an extensive amount of human experience that might be able to be leveraged early in development. But the important thing to remember is that no matter whether you're a botanical or a small molecule, by the time you get to phase 3, the standard requirements that are true for any drug product that you intend to develop are in place. And that can actually be a really difficult time period in development, because at that point we need to have the standard type of toxicology studies to help appropriately inform the monitoring and the understanding. And you also have to have a really good understanding of the material that you intend to study, not only to ensure that the toxicology data that we have available to us is adequately predicting the safety of the material that you intend to study in a phase 3 clinical trial, but also that you have the ability to be able to reproduce that material consistently so that the results that you get from a phase 3 study can be applied to the product that ultimately would be marketed. And there's a lot of challenges to that. The guidance goes into a lot of really good detail. There's also opportunities to have meetings with the agency to discuss specific concerns for specific products that will also, I think, help people, guide, the studies that would be necessary to move toward a marketing application for a new drug product.
Excellent. Well, Dr. Taylor and Dr. Mellon, we have a question from the audience, which is the FDA considering cannabis-based preparations or products nutraceuticals or just botanical products?
So as we just said, once you come in here, you could buy something wherever you want. It doesn't have to be cannabis. You could be studying a dietary supplement. If your intended use is to treat, cure, mitigate, or diagnose some indication, then you have a pathway at FDA to pursue drug development. It does not have to be cannabis. It really does depend what your product is. That's why Dan was just saying you do have the opportunity to have what's called a pre-IND meeting. And we do encourage those for folks who are new to interacting with FDA. Once you determine what your indication is going to be, you can go on the FDA website to the Office of New Drugs and look up all the different divisions and figure out kind of where you might fall. You would email that division and say, hey, I want us to have a pre-IND meeting for my indication. And then once your information comes in, FDA will make a determination. Is your proposed drug product a botanical? Is your proposed drug product a single molecule active pharmaceutical ingredient? Is your proposed drug product a whole plethora of different categories that is determined by FDA, mainly our CMC staff? And then we can provide you with the guidance on what you need to do. But the process is the same regardless of if you are a botanical or not a botanical. You need to submit an IND to the FDA. So the answer is yes, we do look at all of these different things. We can't give you any specifics because all that information is confidential, but there are multiple submissions across the board for botanicals and non-botanicals, and there are pathways for all of you to pursue drug development if that's what you would like to do. Dan, anything you want to add?
I don't think so. Very well said.
We have another quick question from the audience, and I do want to get to some of our other guests too, because I have some questions about dosing and real world evidence too that I think Mara and Vicki could address. But one question from the audience, and Dr. Taylor, Dr. Mellon, see if you can answer this. What additional controls would you expect for a smoke drug candidate versus oral or vaporized?
Dan, I can get started and then hand over to you. If you are intending to utilize cannabis or some sort of cannabis product along with a smoking device, we actually have a center for devices. They're not on the call today, but you would need to also, in addition to all of the controls for your drug product, you would also need to provide controls and a plethora of information about the device that you plan to use. And if it's an approved device, you provide that information, and our CDRH colleagues will advise if there's information that's missing. So that's the first one. And if there is an oral product, there is a long list of categories of different types of oral products, and our CMC experts would be able to help guide you once you enter either with a pre-IND or with an IND, and to let you know what type of information you would need. There are many different guidances about all the different orals. Just think to yourself, if you go to the CVS, how many different over-the-counter Tylenols and Advils are available to you, there are even more categories than you're seeing there. So Dan, I'll give you a moment to add on to that as well.
Sure. I think it's a great question, and a question that we struggle with all the time for not only a botanical product, but for any drug product. If you are going to propose a change in the route of administration, one of the review exercises is asking ourselves, what is it that we know about the material? What is it that we know about the existing safety information? And can we leverage that information to inform the safety of one route or another? We could make the argument that from a systemic safety level, they may be very comparable, but you may have differences in metabolism. You may have different local tissue effects that would be, would have to be addressed. Oral and inhalation, you get different, tissues exposed to different concentrations. We would look very carefully at all that information and try to make a safety assessment to determine whether or not it would be appropriate to proceed with a clinical study. And we can provide additional information, depending upon the specific program, of course, on the types of studies, additional studies that may be needed to try to bridge the existing knowledge gap. And as I noted earlier, the way the botanical guidance reads is it does give a bit of flexibility on some of the toxicology information if we can appropriately bridge to that human experience. But it's important to note that a lot of the toxicity studies that are done for any drug development program are being done because you can't really do some of that assessments in human studies. For example, information to inform Section 8 of drug product labeling describes reproductive and developmental toxicity effects. That's not data that can be readily obtained in a clinical setting, and as a result, those studies are completed to appropriately try to inform labeling and help practitioners appropriately prescribe the product. Those are types of studies that would be necessary to really understand and inform that labeling. So again, it really depends on the specifics. One of the things that the Pharm Tox team also does is try to understand the safety of anything that may be coming out of a device. A lot of these devices may have heating elements. They may have, various materials that are part of the container closure system. All of that is part of the evaluation that is done for any drug product and would be done as well for botanical products.
Well, thank you. This is probably a good way to transition into sort of the next topic for discussion here, which is, and maybe Dr. Seyfert-Margolis could address this one for us. What's sort of the difference between how an agency like the FDA would value real-world evidence versus a clinical trial? And maybe explain a little bit about what those two are. And then we've got a couple of specific questions from the audience that our two guests from FDA might be able to address after we sort of set the stage here about real-world evidence versus clinical trial.
Well, first of all, I recognize that most of you on the webinar probably really want to hear from the FDA directly. And I also recognize as an ex-FDA official that there's a lot of care that needs to be taken in what you're going to be able to hear from these FDA officials about any specificity on any product. So I think that, we should be sensitive to that. I'm seeing a lot of questions coming in about, well, would you be interested in working on this or this technology or that? And I don't think you're going to hear any clarity on those answers. So because they can't give you any specifics on your particular product. I think it's also important to note that what we're talking about is a world of drug development, which having now been engaging with the cannabis industry for about 2 years now through CFCR, I know seems somewhat onerous and scary to people, but it is what it is, I guess is the best way to say it. So on the real world evidence topic, I think there's a lot of confusion about what constitutes real world data versus real world evidence. And there is an important distinction between real world data and real world evidence. So real world data can be anything that you're out collecting on anybody under any circumstances. Often in the world of drug development, it's mining of electronic medical records to see if we can find information about a condition or about a drug and how we may be missing safety signals, for example, and FDA spends a lot of time on that. And when I was at the agency, I spent a lot of time on, large data mining exercises. And in those cases, you often get correlations, but not causation necessarily. And that's a very important distinction between what is causative versus what is correlative. Similarly, when you're looking at what is the level of real world data that may be sitting around with respect to the extensive human use of this product in various forms, we have to keep in mind that that data is not done under a set of controlled circumstances routinely. It may be human experience that's captured from observational studies. It may be human experience that's captured from databases that exist in some of the companies. But the question is it being done under well-controlled circumstances? Vis-a-vis, does it constitute evidence as opposed to data? And there is a big difference. I also know that the concept of randomized controlled studies is very important. But again, how randomized controlled studies are done really can be different in terms of the interpretation of the data. And so there are a number of you I know who have done randomized studies. Much randomized controlled trial work is done in the world of supplements. That doesn't mean it's meeting the evidentiary standards that are required for safety and efficacy assessments for labeling, as Dan and Cassandra were talking about, in the drug environment, which has a much higher level of evidentiary requirements, namely clinical assessments, assessments around the indication that you're trying to use the drug and demonstrate efficacy for, and extensive safety. And capturing real-world data from observational studies without well-defined experimental plans and hypotheses is challenging and often not as substantial in terms of how it's viewed as evidence as safety data carefully collected in the context of randomized controlled clinical trials with clinical assessments as part of that data. And I guess one last thing I would say on this point is there are instances where the FDA has looked at what's called historical control data, where there is, or are natural history studies. And I think Dan and Cassandra were pointing to that as well. And those things may be very important and may be considered for early stage drug development. And I'm just going to be 100%, clear on this. There is a huge difference. And I think in case it wasn't fully heard, there's a huge difference between the level of safety evidence that you need to do a phase 1 or a phase 2 study than for a phase 3 study where you are actually trying to bring a product to market. These are very big differences. So my sense is that there's a lot of value in the real world data. There's a lot of hints, but we have to be very mindful that real world data does not equal evidence that is acceptable or meets the evidentiary standards for approval of products.
Thank you for that. And Dr. Taylor, Dr. Mellon, a question from the audience that sort of touches on this is, and let me know if you can answer this. How does FDA value real world evidence in the cannabis botanical drug development pipeline itself? If you're able to respond.
Well, I can certainly start. I think it's important to stress that both Dr. Taylor and myself are not the clinical review team who would address that. So I don't think that we're the best people to be able to respond to that. I will state that I think there are 2 guidances that were just published last fall that are in draft format that discuss real world evidence. And so I would encourage people to pursue that. And again, I think if people are interested in trying to pull together a development program and have specific questions such as that, then that's the wonderful opportunity of having a meeting with the agency that will help discuss some specifics about any particular application. But I don't think either one of us are the best people to be able to respond to that question.
I concur, Dan. I echo that if folks have really specific questions, and I'm seeing a lot of them in the Q&A, Dan and I really cannot answer them because we don't have enough information about what you're proposing that we can give you advice. And all of that advice is always confidential. It's between you, the sponsor, and us, the FDA, and no one else. And so we would just like to reiterate that unfortunately there's going to be some things we just can't give you specifics on. So we do appreciate Vicki making that clear for us, and we're trying our best to give you the information that we can. Thanks, Sarah.
This is Vicki. I think the question about real world evidence, again, Sarah, is that these are evaluated on a case-by-case basis, but again, making a distinction and in the guidances, and I'm very familiar because I was involved in some of these issues when I was at the agency, when the story of real-world data and real-world evidence started emerging and became part of the regulatory science initiatives. Again, it is very important for us to understand the distinction between data that exists that is either experiential, anecdotal, and collected as part of sort of surveillance or survey systems versus data that is collected in a standardized fashion, specifically with the intent of forming a body of evidence to support a specific point. So it's one thing to say that, oh, you know, we have 10,000 people who've been using our CBD and nobody's had a problem. It's another thing to do a study and say, we're going to set out to observe 1,000 people using CBD under a specific dosing regimen at a specific concentration with a specific understanding of the type of people that are using the product, and to collect that information in a systematic way with care and consideration across multiple components, some of which probably will have to be clinically assessed because of some of the information that already exists in the literature with respect to some of the potential safety considerations of this product. So that doesn't mean surveying people and everybody feels fine and they haven't been to the doctor. That's very different than, you know, a careful analysis, let's say, of, you know, blood draws monthly to look for specific changes in liver enzymes or in kidney function or something else, or how fast these products are metabolized or excreted. So, you know, those are very important components that go into the assessment and what I would call creating real-world evidence as opposed to just capturing data.
Well, with that, Mara, maybe you could jump in on, Vicki mentioned dosing. And Mara, could you inform us a little bit of how dosing is established?
Right. Thank you. I'd like to actually build on what Vicki was talking about as well, because I think that what we see a lot of is people seeing real world data or anecdotal data and making assumptions based upon that to move forward and start making medical claims. And that's the part about the cannabis world that's so frightening. For me anyway, it's like people say, oh, I gave it to my brother and it did this, and now they're going to start treating patients because we don't have the type of controls in place for what people can say and do, which they should. So when I first started working with cannabinoid-based medicine, my goal was to figure out how to accurately dose it for various diseases so that physicians felt comfortable guiding their patients. I still am of the opinion, after some of the research that we've done, that the entourage effect is so enormously important. Some of the research that we've done on that in the past with preclinical, looking at whole plant extract versus single molecule, and the difference in the efficacy on all three major subtypes of breast cancer was phenomenal difference. How much more important it was to have the whole plant. Now, as far as dosing, initially we help a patient to titrate on a set of cannabinoids where there is some evidence that's been done, whether it's preclinical or clinical evidence. Either way, we know a lot of the values. We understand some of the way the mechanisms of action within the body, of how a THC activates a CB1 or CB2 receptor, how the CBD works on these receptors as well, where they work within the body. So we have a pretty good starting point on what to work with when we're selecting the appropriate cannabinoid-based medicine to treat a patient or to help a patient. We were controlling the amount of medicine and how they took it prior to Prop 64 passing in California, which completely changed any kind of data collection efficacy at that stage. But before that, we were controlling with lab-based or lab-tested medicine, where we knew the cannabinoid terpene content for the most part of a medicine, and were able to help a patient titrate, figure out what their therapeutic level was based upon things like blood work and scans and biomarkers and other evidence to show that it was actually affecting, not that they just felt better or they liked the strain or whatever it was. Keeping it really much more on a how's it working at a metabolic level. And then based upon that, may make adjustments up or down on the types of products and the dosing that they would have. And then continuous the feedback loop. When you get enough of that dosing information on a particular, enough data points on a particular a diagnosis, tracking the ICD-10 codes and things like that, you're able to then make some assumptions about what it looks like as a therapeutic dose potential, so that you have a starting point to work against. And the entire purpose of this is collecting enough data points that you can attract the serious research of the clinical trialing that needs to go on. The hypothesis I've always worked from is whole plant medicine is better than single molecule. I'm a strong proponent in cannabis staying as a nutraceutical. I believe that in order to dose it, there's a lot of more forgiveness in it than there is in some of other compounds, because of the fact that it doesn't activate the dopamine receptor in the brainstem, so you're able to have it as a much safer drug without as much risk of overdose.
I just wanted to jump in and so in all of the dosing and everything that Vicki and Mara were talking about, kind of ties back to the endocannabinoid system, right? Because everybody is built a little different with their own genetics, right? So the importance of the clinical trials really does establish exactly that, right? Is a dose safe for everyone? Are there the genetic differences that we see in humans that we also have to take into account, right? So again, it gets back to the complicated nature of both the plant and the system in which that plant plays in our bodies. So.
And we had a question from the audience too, which I'll throw over to our guests from the FDA. What are the current safety concerns or sort of measurable endpoints for cannabis mixtures that we know of at this time?
So I can start, Sarah. So just so we're very clear with everyone, there are only 4 approved products that are related to cannabis at the FDA. 3 of them are synthetic products. That's Marinol, Syndrose, and Sesamet. Those are THC synthetically derived products. And then there is Epidiolex, which is a highly purified CBD that originated from the cannabis plant. There are only 4 approved products. So within those 4 approved products, there are a long list of safety concerns, and they're different based on the cannabinoid. And I'm sure Reggie may be able to speak to this even in much greater detail than I can based on his work. But I think the challenge is, and many of you on the phone know this, that there is interest in looking at other cannabinoids. And since there are no approved products, and I think the message has been very clear today, if there are no clinical trials that are looking at the specific cannabinoids, either individually or as a mixture, it is very, very challenging to gain understanding on what the endpoints would be, what any of those safety concerns are, because if they're not studied in a controlled environment, such as a clinical trial, it's very, very hard for us to gain understanding of what those safety concerns could be. So we have a list of safety concerns for Epidiolex as an example. Liver toxicity is one of the ones that's on the label, but it's important to note that Epidiolex is specifically for pediatrics who are very, very ill and they're under the care of a physician. This is not something that's just readily available to everyone, and they are being monitored regularly. So I think the unknown of all these other cannabinoids, either individually or in conjunction, can lead to a long list of safety issues that are identified and many that are not yet identified. Dan, do you want to add some more to that?
Sure, I can elaborate a little bit. I think that, as a toxicologist, one of the things that our discipline does is we try to leverage the existing knowledge, or we generate data in a non-clinical study to try to inform what those safety concerns are. And as Cassie mentioned, to date, there are 4 products that, with the exception of Epidiolex being a highly purified, are synthetic molecules. And they all went through a very detailed development program to try to predict the types of adverse effects that could occur to inform the clinical studies that were ultimately done. One of the things that in the farm tox world we do is we look at what those exposures are in the animals. We try to extrapolate that, initially with body surface area comparisons, eventually looking at actual exposure data, Cmaxs and AUCs, to try to predict what types of dosing, that would occur in humans and what the potential adverse effects are, and then do the study in individuals. We do that when we feel like we have appropriate safety information to be able to do it appropriately and to inform that safety profile. We have not approved a cannabis product. I think it's important to stress, you've heard, I think everybody has heard multiple times today, that, if there is indeed such an entourage effect, or there is indeed a very different response to a complex mixture than isolated compounds, that is what needs to be evaluated so that we can appropriately inform the safety of any clinical study and appropriately evaluate the efficacy of that mixture. And, I think that's an important point to stress, that is exactly what the data would be needed to undertake a standard approach to ensure this, as best we can, the safety of anybody who would be enrolling in that clinical study.
Could I add one more thing that I think is an important point, Sarah?
Go right ahead. Yeah, please.
So I think the other important point about Epidiolex and these approved products also has to do with, the job of the FDA is to calculate the risk and the benefit. And so as the 2 FDA panelists have been pointing out, safety considerations are paramount no matter what, but the tolerance for safety signals, let's say liver toxicity, is going to be very different in individuals who are deriving a benefit who already have a serious illness, as opposed to what the tolerance would be for liver toxicity in a more broadly used product, let's say for treating a more minor condition, maybe, insomnia, which is something I hear a lot about. So that risk tolerance will be much lower in the treatment of an area where the benefit that's being derived or the condition is not serious enough that it would warrant the risk of liver toxicity. So these are all important considerations when you're thinking about the indication, as the FDA panelists have described previously. It's not just about the safety signal, but it's also about the tolerance for any adverse events depending on the condition you're treating. And so I hear this also a lot from folks in this space who are very interested in things like insomnia or stress or anxiety. And the severity of the condition will also dictate the level of adverse events or safety signals that would be tolerated. And so that's another very important point.
Thank you, Vicki. And I'll leave space if Cassie or Dan want to respond at all, or we can move on to another audience question.
At some point.
Well, we'll give others some time. We've been very popular, Jack, but thank you.
You have, and we really appreciate you taking the time to join us today. Mara and then Reggie, an audience question. How much do you think the role of the endocannabinoid system in the placebo effect impacts the way patients experience the entourage effects, especially in the context of genetically irrelevant product labels like indica, sativa, and hybrid?
Well, that's a very good question. Reggie, of course, can talk to the bioavailability of various terpenes upon method of ingestion much better than I can. However, I've done some very, very small experiments with patients, giving them whole plant medicines versus giving them single molecule, or even single molecule where it's been had an addition of the top 2 or 3 terpenes that are ordinarily included in that particular cultivar or medicine. And there is a tremendous difference in their response. And I think part of it has to go back to the fact that we still don't know what we don't know about the plant. When I first started working with cannabis, we knew about 80 compounds within the plant. Now there's, and I think it was 120 terpenes. Now we know of over 500 compounds within the plant. So I always feel like it's the key log that we don't know what it is. But as far as a placebo, there's evidence of people that have applied a topical to the top of their head and talked about how stoned they were. Not very likely. However, there are instances where if you're treating somebody who's, in their 80s, for example, and they use 1 milligram of THC, and it's in a compound that also contains myrcene and linalool, it might make them sleepy if you give them the same THC dose, but it's in a compound that's alpha-pinene and limonene. It may make them feel agitated or upbeat, depending on how they react to those particular terpenes. So I think in a lot of times, some of the work that I've been doing all these years is these patients don't know enough to have a placebo. They don't have enough information to even assume or think. And so it's not like you're, giving a penny to a child with a wart and say it'll get rid of it. They don't know that this is something that should be happening or should not be happening. And it makes it very clear oftentimes that we have to change the profile of the medicine based upon something is even on the very, very low doses. So I do not believe that it is placebo. I believe obviously anything could be placebo to a certain effect, but I do believe that that's actual science. I mean, terpenes come from other medicinal plants as well. And so why wouldn't we assume that they have medicinal benefit when they're given in a form that they are bioavailable within the body? Not to Reggie.
No, that was a beautiful response, right? And I would add that the concept of placebo for cannabis is a difficult one, right? Because generally we're looking at the cannabinoids for their effect, right? But the terpenes, as Mara said, have their own complete effect. And I would like to remind everybody that the timing difference between Meshulam's paper and another group's paper was like 8 months, right? And so had the other group that was from Italy published first, we might be referring to this as the endocaryophyllene system instead of the endocannabinoid system, right? Caryophyllene binds just as well in that receptor, right? So we really do have to be careful about what we're talking about as placebo. Are we talking about a no terpene and no cannabinoid, product, or are we talking about a no cannabinoid product? Because then we have to expect that the terpenes will in fact have some influence, right? And we know this from all the studies that have already been done in mammals and other organisms, right? Even insects, right, respond to terpenes, right? So there are very powerful physiological effects that terpenes elicit, and they are part of this entourage. And that's why I kind of said before, terpenes are the setup, and then the cannabinoids come in and are kind of the finisher, right? So we really do have to think about this plant and how we go about categorizing things a little bit differently, right? And then you add to that, as I said before, everybody is built a little different because of their own genetics and the distribution and deposition in terms of number of these various G-coupled protein receptors that are now part of the endocannabinoid system. So, sorry, didn't mean to blab there.
Yeah, maybe you could talk a little bit, both you and Mara, too, and actually all the guests here, about what we know about sort of how these are metabolized too. And, you know, in that system, whether a variation of the delivery method will matter. Mara kind of briefly touched on that, but just to maybe elaborate a little bit.
Yeah, so the method of ingestion is very important on not only the metabolization and the uptake as well, and the timing. For example, if something is, if a cannabinoid-based product is ingested as a gummy bear or something, it processes through the body, goes through the liver, ingested through the intestines in a very different way than if something is in a buccal or sublingual as through the mucosal layer. And inhaled, of course, it goes all directly to the brain. There's some really good charts I have on showing the timeline on bioavailability based upon the method of ingestion. And so my experience has been using something that's either a nasal cannula of some sort or as a spray or having it as a buccal is going to be, or a sublingual, are going to be the most consistent in the effect. Because the problem with eating as an edible, it depends on when's the last time you ate, how hydrated are you, what kind of, have you get enough sleep, what is your mood you're in. Everything about that's going to impact the uptake, the speed, and I think that's one of the reasons that we have so many instances that we report. Nobody reports to the emergency room because they smoke too much necessarily. They're coming there because they're ingesting as an edible too much. And normally it takes them 90 minutes to take effect, but now it's been 3 hours and they still don't feel it. So they take more, and then they take an uncomfortable experience results. And that's because of the inconsistency. Now, a lot of products are being nano emulsified now to make them have rapid onset, and also to make some of these liquids and whatnot so that it suspended. It doesn't, people talk about water soluble, and it's not water soluble. It's just suspended in water, really, in these little nanomolecules. And we need to not be making products that we're putting out into the world without having more safety checks on them because of the rapid onset. If somebody is on a pharmaceutical, for example, and I know this is slightly off topic, but I think it's important.
It's not. It's on
not
topic.
Important.
No, it's
Okay.
not topic. It's absolutely on
Totally
topic.
on topic. Keep going.
So if somebody is making these products and your company is being guided by CPG instead of by scientists and medical doctors and whatnot, and you come out with this product and you claim, oh, in 5 minutes you're going to feel it and it's going to wear off in an hour and a half. If a patient is taking a time-released product that is, it can have extraordinarily serious repercussions on speeding up the metabolization of that other pharmaceutical. And then you have an adverse reaction that looks like it's coming from the other drug, but it's really coming as a result of this nanoemulsification.
And I would add one more thing, right? So further, there is evidence that CBD does in fact have a role with cytochrome P450s and may slow down their kinetics, right? So further complicating the situation, because now there are potential contraindications with other drugs because the expected clearance of those other drugs may be affected, right? So there's, again, there's a lot we don't know.
We'll throw it
Anyone
up.
else want to weigh in?
Well, I think those two discussion points were really super important, and also goes back to this real world data, real world evidence question, because again, without systematic capture of data with intent in a certain population, it's understood what they are doing and what other medications they may be on, as was just discussed, it will be very hard to know what the actual profiles of these products are. So these studies need to be done with intent. And I think there are ways that we can start to develop foundational safety data in this industry, starting with healthy individuals and maybe some conditions, but we need to be very careful and systematic about the data capture and data collection for just some of those reasons that were illustrated right there.
Terrific. And now we're at the top of the hour. So I want to thank everyone for joining us today. As you're logging out, there'll be a brief survey. We'd be very appreciative if you'd fill that out. I want to thank all of our guests today, particularly our folks from FDA. I know we did not get to all of your questions, but they've seen all of your questions, and it's going to help inform their process moving forward, I have no doubt. Our next webinar will be held on Thursday, March 31st at 1 p.m. Eastern, and we'll be talking about a recent CBD study on anxiety and the work being conducted by Radicals Sciences and Charlotte's Web. We'll be joined by Palin Thorogood, Dr. Jeffrey Chen, and Tim Orr.
And I can't thank everybody enough for joining us today. And Dr. Taylor, Dr. Mellon, I hope you'll be back to join us again soon. We'd love to have you as guests again. I think this is a really informed conversation. As Jack mentioned, our next webinar is going to be Thursday, March 31st, and you can go to www.uscfcr.org/events for more information. Pre-registration for that will open in just about a week. And also a video copy of today's webinar will be posted on our website. That'll probably go up on Monday, so you can share it with anybody who was unable to attend. And that website again is www.uscfcr.org/events. And with that, thank you all for joining us. This has been a great conversation, and we hope to do it again soon. Thank you all.
Thank you.
Thank you.
Thank you.